Okay, hi everybody. It's my pleasure to introduce Raul Andino, who is a professor in the Department of Microbiology and Immunology at the University of California, San Francisco. And Raul Andino got a master's degree in biology and then PhD in chemistry. And then he worked in the University of Buenos Aires. And after that, in the Whitehead Institute of Biomedical Research, and then at Rockefeller University in the lab of David Baltimore. And now he's working at the University of California, and Raul will deliver a talk about this very interesting, very exciting story. So let's.
Thank you very much. Is this? It is a microphone, or is for the record? Is okay, or somebody in there. Okay, can you hear me well? So yeah, again, it's a pleasure to have been invited and to come here to visit a beautiful city, Moscow. Seeing familiar faces now. I have friends among the students. Yeah, so the story I wanted to share with you today is about this comparative immunology we've been doing, and basically it's at two levels. First of all, we want to understand the logic or the general principles of immunology in general. How is that the antiviral defense mechanisms or bacterial defense mechanisms work to protect the host? And there must be general principles in which mammalian systems and plant systems and insects have in common. So that's one level, and the other level is a comparison between different animals of the same kind. In this case, arthropods. And of course, as we will discuss later, mosquitoes are important organisms, probably the biggest animal killers in the world, because they transmit these pathogenic arboviruses, and like a dengue, Zika, no coronaviruses, but we were discussing earlier today, maybe also coronaviruses. And so, because we want to compare what the best known immune system is in vertebrates, I wanted to compare that, start the lecture by comparing these two. But one thing it's important to say is that we've been focused, of course, in mammalian systems because we are, vertebrate systems because we are vertebrates, but everybody else, every other organism in the face of the Earth, it has to deal with pathogens, but they don't deal using they don't work using a protein-based immunity, but rather a nucleic acid-based immunity. But here I showing you what we know on this vertebrate immune system, where there is exquisite specificity. This adaptive immunity, you acquire this information when you've been exposed to pathogen, and you create specific molecules that recognize very well pathogens, very specifically pathogens, Zika versus dengue, or polio versus Coxsackie virus. And this is a protein-protein interaction. This is an antigen-antibody interaction. But also the other characteristic of the system is a systemic spread. If the immune system can be effective, it has to be one step ahead of infection. And one way to be one step ahead of infection is that the immune system is trained where the infection is happening, but then that response happens in rings of protections or maybe even systemically. And then finally, memory. Many of these organisms live long, 40, 70 years. And so the experience of one pathogen is transmitted over time by creating a new gene, like in this case, the BDJ recombination create this antibody gene. So this thing is slow, or maybe I don't know how to handle this. Let me see. No, I don't. Oh yeah. So as I said, in the nucleic acid-based immune system is in many cases is RNA-based, and for example, and this is the classical, what we call the classical RNAi pathway, where two endonucleases, dicer1 and EGO2 or RISC, participate in this pathway, but there are many other proteins that also play a role there. And the basic idea is that viruses produce an intermediate or double-stranded RNA that is recognized by this enzyme to produce small RNAs, which are then formed this assembled in this complex, which now go and look for a specific nucleic acid. Yeah, I see what I'm saying. What it does is it finds a perfect match between the RNA target and this guide RNA. But there's much less, or was much less known about the systemic spread and memory. So we didn't know when we started this work more than 10 years ago. And I'm going to be here and handle it from my computer, because this, obviously I don't. Okay, so what this has been for many, many years, and what we want to focus on is in this particular type of cell types, and we're going to go through this pathway or model that we've been created. What is going on with this? Maybe this is not the right presentation. It's jumping. Let's see. I have this perfectly organized. Let's see. Okay. I'm telling you, this is okay. So the original idea was that that you have an infected cell, and somehow this infected cell sends signals to the other cells to create these rings of protection. And we thought that maybe double-stranded RNA was an important molecule that could be doing that work, because what happened is we did a screen that yield several proteins, a genetic screen that yield proteins that were involved in the uptake of this double-stranded RNA into cells. So basically the screen was that we take these Drosophila cells, put double-stranded RNA on top of that, and the double-stranded RNA was uptake by the cells to create an antiviral response, to create an RNAi response. And then we look for mutations, actually, RNAi from RNAi. So we use, we do downregulate genes that now destroy this ability. The important sort of observation was when we look at the effect of these three proteins on the ability, on the survival of the Drosophila. So here the Drosophila is challenged with Symbis virus, and you see that these three genes really reduce the ability of the virus to deal with this infection. Very, very similar. If you knock out one of these critical endonucleases in the RNA pathway, you see the same sort of hypersensitive to infection. In the context of those studies, we did this experiment in which basically we vaccinate the flies. So we took double-stranded RNA, injected into the flies, and then challenged the flies with Symbis that carry GFP. So if you don't do anything to the flies, you see the flies became green because this GFP that is carried by the virus. If you put double-stranded RNA from a different virus, this is Drosophila C virus, still the infection progressed without, like you haven't done anything to the flies. But if you put double-stranded RNA against Symbis, a specific, two different specific pieces, you completely abrogate infection. You can see it also by Western blot. Now, there was one thing that it was very intriguing when we did this experiment. and that was how much double-stranded RNA you need to use to create this immunity. There was only, you only need 10 to the 5th, 100,000 molecules of double-stranded RNA. So this vaccination was very, very effective. And that inspires us to think that perhaps there was an amplification step. Similar to happened in vaccination with us, we put a little bit of an antigen and something is amplified. B cells proliferate and now you have a lot of antibodies against the antigen. And so now is the slide that I jump ahead of me. I want to concentrate for a minute on these cells called hemocytes. Hemocytes are the only cell that circulate in the hemolympha, in the blood of the insects, and they are like a macrophages. And so the idea was, okay, is this hemocytes play any role in the immunity? It turns out that if you disrupt the hemocytes by either expressing specifically in the hemocytes, this HML is a specific promoter that expresses specific RNAs or proteins in the hemocytes. So we can create this transgenic Drosophila which express Reaper, which is a pro-apoptotic protein. And when you do that, basically these cells disappear from circulation. You create sort of an immunodeficiency state in Drosophila, and Drosophila is hypersensitive to SIMBIS infection. And also, if you knock down AGO2, remember AGO2 is the second endonuclease in the pathway, specifically in the hemocytes, you also have hypersensitivity. So there seems that the hemocytes have a role in protecting from infection, but also the RNAi system was important. It's not a non-specific immunity, but it's something that is mediated by RNAi. And this is just survival curve. Again, we see that these guys without hemocytes are hypersensitive. Let's see. So the idea then was that perhaps the double-stranded RNA may be uptaken by the hemocytes, and some of that produce primary small RNAs, we call viral small RNAs, and some of them can be amplified somehow to create secondary small RNAs. So this will be derived directly by endonuclease processing of this, and this could be produced by some amplification. This is to address the question why you need so little amount of those RNA. Remember, there are millions of cells in the fly. It turns out that the secondary RNAs, we can distinguish these two because this guy has 1 phosphate in the 5' end, and this guy has 3 phosphates. Why? Because endonucleases, when they cut, they leave, because they cut endogenous, they leave only 1 phosphate. But if these guys are part of the amplification, they might be part of a transcription process. And when you have a transcription process, the first nucleotide has 3 phosphates, right, that accept the next nucleotide. And so we can distinguish by using a special way to prepare these small RNAs libraries, that we can distinguish those small RNAs that have 3 phosphates from those that have monophosphates. And now we took the flies and we separate the hemocytes from the rest of the flies. How do we do that? We develop this bleeding process. Basically, we chop up the head of the flies and put them upside down in an epidermis tube, and make a little small in the epidermis tube and spin the epidermis tube in another epidermis tube. And then we bleed them that way. It's not approved from human use, but it works out for this Drosophila. And then you can further purify the hemocytes. But the important result is here. So here we're looking at different types of small RNAs, microRNAs, piRNAs, ecRNAs, endogenous small RNAs, and viral siRNAs. These viral siRNAs, what basically, all of them, the way we identify what type of RNA is by sequencing. right? So we know this is a microRNA, and we know that this is a VSRNA because it has sequence that corresponds to the viral genome. And what you see here is obvious. When you look at the hemocytes, most of the viral VSRNAs are 3-phosphate RNAs. So they are these kinds. And all of them, you only see a little bit of these 3-phosphate, but all of them, most in carcasses, you don't see them, but all in hemocytes. So it suggests that the hemocytes has the ability to amplify this signal by creating these secondary siRNAs or VSRNAs. But how do they do it? And here, it was an observation from a former postdoc in the lab, Carla Sale, that she was studying persistent infections in Drosophila cells. And what she found is that the cells that actually maintain viruses persistently infecting cells, cells that are persistently infected by viruses, they create a DNA which is product or has the same sequence than the viral RNA. So we said, because in plants and in C. elegans, there is an amplification of the viral RNA by RNA-dependent RNA polymerases. But in Drosophila, there is no such an enzyme. So we look at whether, when we infect Drosophila and isolate hemocytes, can we just PCR using specific primers corresponding to the virus? Can we PCR what we call viral DNA, right? And we also use this purification to sequence small RNAs and detect how much of these tertiary RNAs, 3-phosphate RNAs are. And here's the results. Let me walk you through. So here is infected or uninfected, and here you have carcasses of hemocytes. When you don't infect it, you don't see this DNA. This is DNA is just PCR. This is the control is one gene. And so you see that there is no carcasses or hemocyte and uninfected. But in infected, you see a little band here that is concentrated only in the hemocytes. Okay, the hemocyte has the capacity to produce this DNA. How is this DNA produced? If you add this compound AZT, which is a reverse transcriptase inhibitor used in HIV anti-retroviral therapy, AZT is inhibitor of reverse transcriptase, and when you do that, that band disappears. So the proposal really is, okay, so one more thing. So then we look at these VSRNAs. Do you produce VSRNAs? And then indeed, if you look in the presence in the absence of the AZT, so in this case, you see production of VSRNAs, and in hemocytes there is more of these VSRNAs. But if you put AZT, those VSRNAs disappear. And then, of course, what is interesting is that even though this is an antiviral compound against HIV, when you put these antiviral compounds, SIMBs replicate at higher levels. So you need to produce this DNA here through an endogenous reverse transcriptase in order to produce these VSRNAs, because here you don't produce it, in order to protect against virus infection. But there's a problem. This process is all inside of the hemocytes, right? And if the virus replicates in any other cell type, how is it this system serves to block virus infection? How is this RNA here can go and target the viral RNA? And then we propose the idea that perhaps these little guys, VSRNAs, are encapsulated in exosome-like vesicles and shed from the hemocytes, and this form in order to deploy and distribute these antiviral small RNAs. And there's some particular proteins that are involved in the formation of exosome-like vesicles, and we knock them down. And what you see in there is that if you knock out RAB1 or 6-1A, in both cases you have increase in virus replication. So it seems that these vesicles somehow are involved, perhaps, in some way of antiviral protection. But the real experiment, the real proof, was in this more complex experiment. What we do here is we have donors flies. So there's some flies that are not infected, so we extract the hemocytes. Now we bleed them, but we remove the hemocytes from the preparation and then precipitate the exosomes. And then we have flies that are infected, but this fly is treated with double-stranded RNA control. This fly is, the hemocytes express reapers, so there's no hemocytes. And this fly is expressing double-stranded RNA against RAB11, so it doesn't produce exosome. And so we purify the exosome-like vesicles, and then co-infect naive flies. And then we measure this particular Symbis has luciferase, so we measure luciferase activity. And what you see here is, this is hemocytes from a naive fly. This is hemocytes from this fly that has normal hemocytes and produce exosomes. So you see that there's a big inhibition. And these are from Reaper and RAB11. So this is a process that in human immunology is called passive immunity. So basically, you have an Ebola patient that recuperates from infection, you draw their serum and take this serum to passive protect or immunize the next patient that is. And so this is the same thing. You extract exosome from one fly and protect the other. Okay. So the model becomes a little bit more complex now. This thing is infected. Somehow the hemocytes get closer to these infected cells and uptake the double-stranded RNA from these infected cells. We don't know exactly what the hemocytes are uptaking. It could be that the hemocytes is engulfing the entire infected cell. This is something that we don't know exactly. We also don't understand right now how is the hemocyte know that this infected cell is infected, but we assume, or the assumption is that the infected cell is sending some signal of danger signal of infection, like interferon signal. But once the double-stranded RNA is in the hemocytes, then endogenous reverse transcriptase take this double-stranded RNA, convert it in bDNA. This produce small RNAs. The small RNAs now encapsulated in the exosomes and are distributed to uninfected cells. So we know now a little bit more about the systemic spread, because systemic spread is given by the hemocyte and the exosomes and memory. The memory is this vDNA. And if you think about it, basically it's very similar to what happened with antibody. The original naive fly doesn't have this gene. It's acquired once it's exposed to infection. Okay, now I'm going to switch to mosquitoes. And what I wanted to do, the reason why I want to switch to mosquitoes is because the mosquitoes give us 2 or 3 pieces of information that are very intriguing and extend our, sort of understanding of the complexity of the system. Of course, there is this other medical thing that I need to put in the grants when I write the grants. This is very important, but also you will see that give us some future directions of. The mosquitoes have a little bit a different flavor because in addition to produce small RNAs that are about 21 nucleotides in length, produce these other RNAs which are a little bit longer, about 24, 25 to 29 nucleotides in length. These are not very precise, these are very precise 21. So you can see here a 21 nucleotide, a small RNAs. Okay, so this is small RNAs produced during Symbis infection, and you see the different sizes here and the amount, number of copies, and so you can see that there's a peak at 21, and then there's a peak between 25 to 29 here in black. Now, if you treat these cells, these are cells infected with mosquito cells, AG2 infected with Symbis, what you see here is that if you downregulate AGO2, you don't produce the small RNAs, the siRNAs, but you don't affect the large RNAs. But if you downregulate this other protein called PW4, now this peak of 26 or 28 or something, these larger small RNAs disappear. And this protein here, PW4, belongs to this family of PW proteins. And actually these things are called piRNAs derived from this family of PW proteins. I honestly think, okay, so I think I have the wrong presentation, but we'll do our best. So one thing we need to know about these PW proteins is that in us and in Drosophila, the PW system is restricted to the germline, and their function is to protect from the mobilization of endogenous retrotransposons. So when a retrotransposon produces RNAs, you produce these small RNAs by this set of proteins, and these small RNAs target the transposons. So it's a way to protect germline. But in mosquito, this family has an amplified. Instead of having 3 proteins, now you have like 7 or 8, or something like 8 proteins or more. And now they create, or the idea is they create this other antiviral function. So we're going to walk through that antiviral function in some detail now. We show that if you inject double-stranded RNA in mosquitoes, those RNA against PW4, and then you challenge the mosquito with dengue virus. This is in midgut and carcasses, and here you're measuring genome copies. You see that there is an increase of replication of dengue in both areas. And also you can measure PFUs, and also you see that there is an increase of virus replication. So this suggests that this PW protein is indeed a restriction factor in mosquito, but we don't know exactly how is that working. And the one thing is interesting is that PW4 is upregulated by blood meal. So, many immunological relevant proteins, they get upregulated when you need it. You don't want to have them all the time. Mosquitoes also produce vDNA. Here you see over time an infection with Symbis. At 24 hours you start seeing something, but then you see very clear 48 and 72 hours. If you treat cells with AZT, now you have an increase of virus replication with Symbis compared to the untreated. And when you treat the cells with AZT or D4T, which is another reverse transcriptase inhibitor, again, you don't have any, you reduce very much the production of piRNAs. So the idea is that the vDNA templates the spike RNAs. But the sRNAs are not templated in the mosquito by vDNA. So if you see the siRNAs are not affected by the treatment with the antiviral. So now I'm going to walk you through a new model, and the model here and the rest of the talk is going to be focused mostly in two aspects of this process. Acquisition of the vDNA, how do you get the vDNA to be created? Where does it end up sitting, this vDNA? Is it works as an episome, that's inserted in the chromosomes, and what happened to them? And then a little bit of a processing, once you have this vDNA, how do you process this RNA to create the antiviral compound, the small RNAs. So first is about this reverse transcriptase and recombination process. So the idea is that perhaps when an infection happens, this RNA is sitting in the cell somehow, and the retrotransposomes produce reverse transcriptase that now somehow can take this RNA and produce a recombinant. So the reverse transcriptase can start replicating in the retrotransposon, but then jump to this template to produce this vDNA. And the way we did study that was by infecting mosquito cells, or looking at persisting infected cells, or SIMBIS acute infected, isolate DNA from the cytoplasm. And because the way that retroviruses replicate, they often accumulate a circular form of their genome. Sometimes this is a dead end product. It's not something that it's used further for integration or anything like that. But so what we thought is, okay, maybe what we can do is extract DNA from these infected cells and purified circular DNAs like you purify plasmids from bacteria. And then you treat this purified or semi-purified prep using exonucleases to eliminate the background. And then once you've done this, you do deep sequencing using this Nextera pair-N sequencing approach. And what you get is this picture. So here are reads for SIMBIS, or for this a persistent infection flavivirus, CFAB. And what you see is that you see a distribution of vDNA sequence all along the viral genome. So there is no biases where the vDNA is formed. This recombination can happen any place on the genome. So this reverse transcriptase can pick up many places, or one or more places of the sequence from the virus. And then what we found also is we look for these junctions. So the idea is that you will have transposons in one side, and at some point the reverse transcriptase jump from the transposon genome to the viral genome and continue reading. And so you should be you should see hybrids, this junction. And indeed we see sequence and start here and end up here. Start in the SIMBIS sequence and end up in this transposon TY3. And then what we also can do by doing this is, by looking at these hybrids, is we can look at what kind of transposons are doing the job. Because now we have sequence of transposons that are hybrid with the SIMBIS sequence. And what we discovered is that these two types of transposons, I mean, in this case, is TY1 and TY3, and this there's another little amount of other transposons, but these two here are the transposons, the one kind of transposons that are very similar to HIV. They are called LTR retrotransposons. Okay, and the replication is similar to HIV. Okay, so now Another important thing in general in comparative immunology is that you want to have a system that protects you against foreign agents, foreign invaders, but you don't want to kill yourself, right? In the process. So it's the idea of self, no self discrimination. So there is a lot of these cellular RNAs floating in the cytoplasm of the cell. How is the reverse transcriptase or these retrotransposons know how to choose viral RNAs versus cellular RNAs? Or do they? Because if they don't do that, you will create small RNAs that will downregulate your own genes. And it turns out that if you measure the vDNAs that are derived from SIMBs, in this case is 6 billion reads or something, or 6,000 reads, I forgot now, 6,000, in this particular experiment. And the majority of the mRNA sequence, very, very low. There's a few mRNAs that seems to be reverse transcribed. They are all lower than this guy, but the majority is 100,000 times much more low than the viral RNA. So this reverse transcriptase system specifically see viral RNA and ignore the mRNAs. And this is one of the big questions that we are working on. We don't have a mechanism yet, but how can this discrimination be so powerful? It doesn't depend on the abundance of the mRNA, so there's many abundant mRNAs that are not are completely ignored by the system, and it doesn't depend also on the length of the RNA. You can imagine that longer RNAs are high probability to be reverse transcribed, but that's not the case. There are some mRNAs that are very long compared to the SIMBs, and it's much lower. So that's a big question. Another big question here is, these hemocytes are producing this system all the time, or there is a regulation? Is any regulation? And I already alluded that the macrophages have to somehow know where to go in the body to pick up the nucleic acids. And we did this experiment asking the question of whether reverse transcriptase is active all the time or is induced during infection. And the result is that, in fact, if you infect Drosophila, wait 2 days, take hemocytes, and then look for reverse transcriptase activity in the cytoplasm of the hemocytes, you see that between MOC and SIMBs, there's a huge induction of reverse transcriptase. So endogenous retroviruses are not continuously being mobilized, only they get mobilized when they are need to be mobilized. And this is, again, one big question for us is, what regulates these retroviruses? How is this retrovirus being now somehow included in the regulation of the immune system? And it's, again, this big thing that we're trying to do now in the lab is identify factors that could do the signal transduction. Here's an immunity, here's an infection. We're trying to understand, what participate on that. Immunological memory, we wait for 21 days, and you see that this hemocytes contain this DNA. So for the life of a fly, is 21 days is long. I don't know what would be 30 years in human life. I think life live what 6 weeks? Life 80 days. Okay, so that will be this is a short memory, but what we don't know yet is that whether you can recall this bDNA. So what we don't know is if you create a memory and you have a secondary infection, whether the response to the infection will be more rapid. This is something that we need to study. So, but one way that you can imagine that you create memory that goes beyond 21 days is that this thing gets integrated in the genome. And of course, endogenous retroviruses has the ability and the enzymes to get integrated into the genome. So one thing we ask is, can we see signatures of this this vDNAs in the genome? To do that, what we did is sequence the entire mosquito, these Aedes aegypti cells, AAG2 cells, and we did this using PacBio, which is a sequencing method that allows you to have a much larger sequence than Illumina. And this is important because normally Illumina will give you 100 to 150 nucleotides in length, and if you get into these retrotransposon regions, there's a lot of repetitive sequence, and then the computer doesn't know whether already has go through this sequence or is reading a new one, because it's identical. But with the PacBio, you got a much longer sequence. So this is a way to penetrate what we call here the dark matter of the genome. And it turns out that Aedes aegypti have 55% of their genome and endogenous retroviruses. Similar to the bats that we were talking the other day. There's a lot of these things that, and our genome also, there's a lot of these sequences, and people who don't know what this sequence are doing, and there's a lot of work. But what you can see here is that there's different circles that represent different type of transposons. In this case, this green is the LTR retrotransposon, DNA transposons, sign, and so on. And so what we did is, if you focus on this area here to get a little bit more sense of how the things are organized, you find this following picture, where each line here represents one type of transposon by color. Here are the LTR retrotransposons, and in general, what you see is that where the LTR retrotransposons live, they exclude other transposons. So in here, there are only these green guys, and the blues and the reds are excluded from that region. And because I already told you that the LTR retrotransposon seems to be the ones doing the recombination and picking up this viral sequence, we were interested in going deeper on that. And what you see here in this region, where most of them are this green LTR retrotransposon, is where you see the piRNAs being produced. So if you try to match piRNAs sequence with this genomic sequence, those piRNAs come from those LTR retrotransposons. If you go deeper into that particular region, what you see is there is a lot of this sequence vDNAs into the genome, and many of them are producing these piRNAs. And now, this inserted sequence has a common name, and it's called endogenous viral elements. And we call them endogenous viral elements, but of course, not all endogenous viral elements is involved in this immunological process, but everybody is trying to understand what they do. This particular one here produces all these piRNAs. They are derived from, this is CFAV, this persistent infected virus. This is a TY3 retrotransposon, but it's rearranged with other transposons. So here there's two different TY3s in blue and green, and inserted between this protein and this protein, I don't know what they are, I think it's the GAC region. There's this sequence of the CFAV, which is this EVE, endogenous viral element. Okay, so that's the structure, but that still doesn't tell you anything how this thing is regulated. The only thing it says is consistent with the idea that vDNAs are uptaken by recombination, reverse transcription recombination process. It tells you that when these endogenous retroviruses integrate in a particular neighborhood, it tells you that these guys are active to produce piRNAs. it also tells you that all the orientation of these endogenous Eves are oriented to produce antisense piRNAs. What I mean by that is that these small RNAs produced here are complementary to the genome of the CFAB, or are complementary to the genome of the virus that has to be targeted. And that is intriguing or interesting because that type of piRNAs are going to be the ones are effective to downregulate virus replication. And the question was, okay, is a PW4 involved in processing? I tell you, I have the wrong. We did one experiment in which we tag PW4, and then we do this APMS, this mass spec analysis, to see what proteins are interacting with PW4. And intriguing, what we see is the PW4 interact with EGO2. So remember that I showed you one of the earlier slides that when you downregulate PW4, siRNAs also disappear. So there is some interesting relationship between the classical RNA pathway, EGO2, and this PW pathway, PW4 pathway, and we don't really understand. But the other thing that from this proteomic experiment emerged interestingly is that there is a lot of factors that are nuclear factors. There are proteins, transcription factors, or this factor here, for example, I cannot read it. It's I forgot now. RXF2, which is a nuclear factor that helps in export and import of RNAs into the nucleus. And also, we noticed that PW4, it shows a nuclear localization when you stain this thing. So this suggests that perhaps the processing of these long RNAs derived from the long Eves to piRNAs may be mediated or facilitated by PW4. And so we're trying to understand that. And one of the things that we did is this question. We try to address this question of whether PW4 associate with these piRNAs, directly associate. And the way we do this is we infect cells, or we look at piRNAs derived from already Eves that are present in the cell. And we immunoprecipitate using antibodies against this PW4 stag. So we immunoprecipitate that, and then we sequence. And what we see, something very intriguing. But first of all, if you look, we can distinguish RNAs or sequence that derive from this Eve and the genome, because this was an integration that happened in the past, and now we're using a new virus that is circulating, it is persistently infecting the cell, another CFAB. So there is some difference in sequencing. So we can look, will be acute infection or persistent, new acquired persistent infection, and these are Eves that were present previously. But the important thing is that when you look at that, PW4 ignore completely the piRNAs that are not coming from Eves. So this is intriguing. It suggests that if you have piRNAs that are accumulated in the cell but have not been integrated, then PW4 cannot process them because it doesn't associate with PW4. The other thing is interesting is that we look at piRNAs associated with PW4 derived from Eves or the surrounding endogenous retroviruses, or retroviruses that are far away and not in the same cluster. And what you see is that there's a big bias of PW4 to associate with Eves, but not with endogenous retroviruses. So there's again a discrimination of PW4 of RNAs that are coming from EVEs from RNAs that are coming from any other place of these endogenous retroviruses. It's complicated, we don't fully understand it, but the point I was trying to make is that there's a lot to understand here in terms of the chromosome organization, how is these EVEs are organized. I mean, what is the chromatin involved? How is this organized? And how PB4 interact with those clusters. So there's a lot to work on here. And I'm almost done. How much time I've been? I think it's up for long. Yeah. Okay, 5 more minutes. There's no much. I mean, this is just ask the question whether the piRNAs derived from EVEs are functional, and indeed they are. You know, these are different EVEs, and this is some EVEs produce more piRNAs than others. We constructed a reporter with Renilla luciferase, and we put the sequence corresponding to these EVEs, and you see that it's a dosage-dependent inhibition. We also create artificial viruses with the EVE sequence, and you also see that when this virus carry the specific sequence is inhibited. So the piRNAs derived from EVEs can be functional, and then finally there is another observation here in persisting infections, and what we do is these cells are persistently infecting with CFAB, and we downregulate now. Well, this is a control, EGO3 or PB4, that are two proteins involved in piRNA production, and you see in both cases when you do that this CFAB increase in replication. So it seems that these piRNAs derived from EVEs play an important role in controlling persisting infection. So just to finish here, one thing is interesting is, I think some of you already noticed this, we sequence a cell line, right? And we find a lot of EVEs that are in this cell line. The cell line was derived from some mosquito. So that means that those EVEs that perhaps at some point were formed in the hemocytes now are germline transmitted. Now they can be transmitted to the next generation and be fixed in these cell lines. So the question really is can this be similar to CRISPR, where you have a bacteria that acquire these repeats and is transmitted to the progeny, creating really population memory, creating mosquitoes that now are refractory to infection. And so obviously they have epidemiological implications, because you can imagine that the acquisition of these EVEs can create mosquitoes, some mosquitoes that are resistant, and then or they are better vector or less better vector. And in some way this is a Lamarckian sort of concept. I know this is maybe the wrong place to mention this, but the idea that experience in life and infection can actually result in that your progeny will have a new characteristic which is resistant to this virus is something that we need to revisit, of course, in the context of the classical evolutionary theories. So we will look actually at many other insects. So this is, many families of other mosquitoes and bees and ticks. And we've been looking, what we've been doing is looking at which ones produce piRNAs that corresponds with EVEs. Basically, those are genomes, and we look at in the genomes which ones have virus-like sequence, and then we try to correlate those EVEs with the expression of piRNAs. Well, first of all, what you would see is in red, all these EVEs are producing negative stranded antisense piRNAs. When you compare the size of these things are slightly different. For example, the ticks here, interestingly, produce two types of small RNAs. One is more like a 21, and the other is like a 26. So what I mean by that is these guys obtain EPs produced by RNAs that are antisense, but the processing machinery is different in different animals. So some produce larger, some smaller. So that will be very interesting. Who is processing really this large transcript and small transcript? And just a summary, what I told you is adaptive immunity, systemic spread, hemocytes and exosomes, acquire specificity by BDNA. These VSRNAs are amplified. And then memory is BDNA and the insertion. The evolution, I think, obviously there is a lot of interest in understanding the interaction between now three elements, right? So you have the host, the virus, and now the transposon. And how is the transposon has been domesticated to increase or facilitate or produce solutions to the kind of things that the host is encountered. So that's a very interesting interaction of three elements. And then regulation, of course, innate immunity is very interesting. Discrimination, self, non-self, is something that we want to understand, like every other immunologist. And then finally, the group that there's one person missing here. Well, okay. So Zach and Patrick. Patrick is more bioinformatician, and Zach became to the lab as an experimentalist, but converted to biostatistician. So those two guys, together with Mark Kunitomi, who is a full-time biostatistician now, they work on the genome, mosquito genome sequencing. They also work on analyzing some of this sequence. But really, Michelle Tassetto has been inspiring the whole thing from the Drosophila all the way to the mosquito. Aravinda was in the lab. Now he has his own job, and he is working in something interesting. I didn't have the time to tell you. That is, of course, the viruses are exposed to these immunological systems, but they try to do something against that and create suppressors of the system. And Aravinda has been working on that, or was working on that. So again, Michelle, Zach, Patrick, Mark, Miguel is, his picture was not there, but he's working on that. I also wanted to thank Carla, who's really started the whole project several years ago. These are our collaborators in PacBio, Ellen and Melissa, they help us with the sequencing. Irma and Ken are from Colorado State University funding, and thank you very much.
Thank you very much. Well, we have probably a few minutes for a couple of questions.
Thanks for running the talk. I have questions about exosomal experiments. A few actually. First of all, have you checked transcriptomes and cytones of these exosomes to see what's inside? Second, if you shut down the reverse transcriptase, how does it change if you have checked it? And how will it affect immunity in responsive flies, which were not introduced to virus? If previous line was exposed to virus but has shut down reverse transcriptase, and if the new line has shut down transcriptase.
So let me start with the first two questions, which I understand, and then we go to the third one, which is more complex. We done proteomics of the exosomes, and there's a lot of very interesting proteins that, because obviously one of the questions is how is the exosome recruit the small RNAs? The small RNAs likely not to be encapsulated or packed in this exosome naked, but associated with proteins. And so we have some ideas of how this happened, but it is not a complete story. We need to study more. And of course, the other thing is, once you have this exosome, how is this exosome targeted to the next cell? is a receptor for the exosome? Is this contained delivered in the other cell? So the idea that we have is that when you inject this exosome, basically what you have is a receptor of the exosome, and siRNAs will be in the next cell. Probably it's not just siRNAs, but maybe it's a complex. siRNAs complex with proteins which extend the half-life of the RNA. But again, that's another question. You know, how long does this last? But the other thing is very interesting that I guess you mentioned is, are these exosomes produced all the time? And one of the models that at least in my mind is that if you remember, we showed that reverse transcriptase activity is increased during infection. So imagine that you have a secondary infection. Now reverse transcriptase is activated, but not necessarily only to produce more vDNA. But if you think that these clusters of endogenous viral elements are in transposon, maybe the whole cluster is activated by infection. And therefore you create new exosomes with more fresh small RNAs that are produced when you have a new infection. I'm not sure if that addressed your question.
And the last question was, if we have shut down reverse transcriptase, the fly won't have this immunity, yes?
Yeah.
But will its exosome produce?
Yeah, this exosome produces still.
Even if this fly don't have immunity, exosomes still produce immunity in other flies, yes?
Okay, so if you we haven't done an experiment. It's an interesting experiment. If you put AZT, you don't produce new vDNA. Yeah. What we don't know is that if in the presence of AZT, whether endogenous viral elements will produce small RNAs still. Presumably, there's no reason why you will not have transcription from those EVs if those EVs gets activated and transcribed. Because reverse transcriptase is not required to make RNA from EVs. Yeah.
Let's have a look from the other side of the problem. Do you exclude the immunogenicity of pure RNA itself?
Yes.
Excluded.
Yeah, it's excluded in one way. Maybe there's something in your mind that I don't capture, but let's put it. I'll tell you the way I think about it. If you immunize with double-stranded RNA against Drosophila C virus, Symbis still replicates very well. So the double-stranded RNA per se is not a. Yes. Right. Yeah.
It didn't work.
It didn't work.
That's strange.
Why? What is in your mind?
Because we can't develop it. And have some experience that it's possible.
It's possible to produce immunity against double-stranded RNA.
Just immunize animals with purified RNA.
Yeah.
Virus RNA.
Yeah.
Cellular viruses, plant and animal.
Yeah. No, I believe you. I believe that in a vertebrate system.
Modified RNA.
Right. I mean, in a vertebrate system. Of course, you will induce interferon, and also you might induce antibodies against double-stranded RNA, and that could have an effect on virus replication. I don't say no, and it's interesting. But in these insect systems, there's no antibody, there's no interferon. In fact, there's no PKR. You know, there's no way that double-stranded RNA will induce this innate immunity per se.
And also, autoimmune disease patients, you know, in blood always.
Yes, it's true. It's true. Yeah, it is true.
Any more questions? So, let's say anyone wants to.
Thank you.